Hello, this group will be presenting on the paper Construction of a Genetic Toggle Switch in E. coli by Gardner. So for the introduction, describe the layout of the two toggle switch plasmids that were constructed and how does switching occur between the two steady states. So the plasmids contain two promoter regions that constitutively express their transcript. Promoter 1 contains the repressor for promoter 2, and promoter 2 contains a repressor for promoter 1. This, in its most simplistic mathematical model, is a caricature of a two-element positive feedback loop where both elements are inhibiting the other. The whole point of this experiment was to systematically study the transcription rates and relationships that can be found in the lambda phage, where it also contains a two-element positive feedback loop between two steady-state attractors that induce two different developmental fates, the lytic or lysogenic behavior. Additionally, we can track the current state of the system by measuring the levels of GFP expression at a given time, since we have one downstream reporter gene known as GFP-mut3 after repressor 1. In this experiment, we can consider the repression of promoter 1 to high levels of GFP expression and the repression of promoter 2 to low levels of GFP expression. The switch between promoters is based on the external cues from inducers such as IPTG, ATG, or heat that can inhibit the repressor used in this experiment. Box 1, represented at the bottom left, is the continuous ODE formalism for the rates of change for both repressors. These ODEs encapsulate the rate of production and the rate of degradation. These ODEs also represent a negative sigmoidal behavior of the two concentrations where the alpha represents the max rate of transcription that will be effectively decreased and dampened by the relative concentration and the binding affinity to the multimeric repressor complexes, which can be denoted at the bottom of V beta and U gamma. The Hill coefficient for cooperativity is crucial for proper bistability, which will be seen with subgroup 1 in figure 2. My next question is, which elements of the plasmid map were used to control the promoter strength? So they used previously studied well-known promoter and repressor pairs, such as the LAC promoter and repressor, the TET system, and the C1 promoter that can be inactivated with heat. They all have varying degrees of affinity and transcriptional rates. So in order to regulate and equalize the rate of protein production, they had to implement new ribosomal binding sites downstream from the promoters. The toggle switch can be seen as an archetype of a biological memory unit, since the basins of attraction in a given system can be associated with a developmental fate in a cell. In a system that undergoes irreversible differentiation, where two stable attractor states are possible, the system has to make a decision and to commit to one fate over the other fate, which can be associated to the process of falling into the right basin of attraction that corresponds given external signals or internal cues.
So my question was to describe the conditions necessary for bistability outlined in box 1 and demonstrated in figure 2. Box 1 just describes what's happening in figure 2 as seen, on the right. Figure 2a and b are derived from differential equations used to measure the rates, concentrations, and cooperativity of repressors in two toggle switches in different simulations. So in 2a, that represents the bistable result, and 2b is the monostable result. To achieve bistability, as seen in 2a, you need three different conditions. The first condition is that you need to know that the cooperativity of repression of transcription are represented by gamma and beta. And the larger gamma and beta are, the more cooperativity of repression leads to a larger bistable region. So in 2D, the one closest to the middle, beta and gamma, are equal to 1, and it's a very strong or a very small region. And as beta and gamma increase greater than 1, you have a larger region of bistability. So beta and gamma must be greater than 1 as the first condition. The second condition is that repressors must be synthesized at the same rate so that it can produce the three intersection points as seen in 2a. So the first intersection point in 2A is seen on the top left, state 1, and then the second intersection state is the separatrix, where it's an unstable steady state, and the last intersection is on the bottom right, in which the top and bottom states are both steady state stable conditions. Um, and the last condition is that you need two basins of attractions, and that just means you have to have two distinct outcomes. And so if one of the toggle lands on one side of the separatrix or the bottom side of the separatrix, it determines whether or not the graph will become bi or monostable. So as again in seen in figure 2A, the separatrix, depending on what side the toggles fall on, just determines what kind of stability it's going to have.
So the question I'm going to explain is, how many stable and unstable steady states are in panels 2A and 2B, and why is there a difference between these two panels? So in panel 2A, there exist two stable steady states and one unstable steady state. The mu on the x-axis represents the concentration of repressor 1, and the V represents the concentration of repressor 2. So one stable steady state has low concentration of repressor 1 and high concentration of repressor 2, while the other stable steady state has high concentration of repressor 1 and low concentration of repressor 2. The unstable steady state lays in between the two stable steady states on a line called the separatrix. And it's unstable because any movement off of the steady state will cause attraction to one of the two stable steady states, or make or be put inside their basins of attraction. Panel 2B, on the other hand, only has a one stable steady state and no unstable steady state. So this whole plot is just a basin of attraction toward this one stable steady state. So there's a difference between these two panels because in panel 2A, the promoter strengths or the rates of synthesis of the two repressors are balanced, allowing for a bistable toggle network, while in panel 2B, one promoter is much stronger than the other, causing a monostable toggle network. This is because if one promoter is much stronger than the other promoter, not only will there be more transcription on that one promoter side, but since one promoter transcribes a repressor for the other promoter, there will also be less transcription for the other promoter. And this will cause this monostable toggle network, or the no clines intersecting only one time. When the promoter strengths are equal though, the no clines intersect 3 times, allowing for 3 steady states.
Okay, so here we're tasked with explaining how the changes in parameters beta and gamma affect bistability. And so first of all, let's start by defining our parameters. So beta represents the cooperativity of repression for parameter 2, while gamma is the cooperativity of repression for parameter 1. And the slopes of bifurcation lines are determined by beta and gamma, and that's the case for large alpha 1 and alpha 2. And we know that higher order cooperativity, otherwise known as a large values for beta and gamma, correspond with a larger, more broad bistable region. And we can see that in figure 2D. We'll jump into that in just a bit. And this makes sense because a larger beta would correspond with a larger slope for the bifurcation line between monostable state 2 and the bistable state, as shown here. And a larger gamma would correspond with a smaller slope of the bifurcation line between this bistable state and the monostable state 1. And since the bistable state is in the middle of the two monostable state, this change of the slope from a larger beta and gamma would correspond to a more broad, larger bistable state, as shown in figure 2C on the left. And from a scientific mechanism-based perspective, it also makes sense because cooperativity would allow for the repressor to bind to the promoter much more efficiently than if there was no cooperativity at all. To be bistable over a wide range of parameter values, the two states must be tolerant to fluctuations that might come up, and therefore the toggle switch should not be able to be flipped randomly in between the states. A higher order or higher cooperativity value allows for much better repression and therefore reduces the chance of a random toggle switch from one state to another. Therefore, since the stable state of the toggle arises from a mutually inhibitory agreement of the repressor gene, a larger cooperativity value will further help the repression and therefore better the mutually inhibitory agreement. And this can all be seen in figure 2D, where a reduction in beta and gamma leads to a smaller bistable region. Visually, this is portrayed here where beta and gamma are equal to 3. We see the broadest bistable region, and then it's smaller for a value of 2, and then the smallest bistable region for a value of 1.1. Thank you.
For figure 4, the first question is, how do panels 4A and 4B demonstrate bistability relative to the controls? 4A and 4B exhibit the difference in expression between the controls, which were PTAC 102, 106, and 108, which are shown in the bottom two panels of 4A and 4B. And the presumed bistable plasmids, which are 4 PTAC and 2 pike, only one of which pike was actually bistable. In 4A, the top panel exhibits the fact that once GFP expression is switched on in the 4 bistable PTAC plasmids, they remain in what the authors call the high state throughout a period of no exposure to IPTG. Then are switch off as expected when exposed to a temperature of 42 degrees Celsius. This is contrasted directly to the controls, in this case being PTAC 102. In which GFE expression is started with exposure to IPTG, but does not continue its absence. The experimenters also chose to display temperature-induced PTAC 106 plasmid in the bottom panel of both figure 4A and 4B by showing that the GFP was expressed in this control. And in this case, PTAC 106 is inducible to produce GFP by a high temperature. The experimenters demonstrated that a temperature of 42 degrees Celsius was reached during the treatment. And then in 4B, it was very similar to 4A. In its setup, the main difference was that the molecule ATC is used rather than IPTG to induce GFP expression in the first control plasmid and the two experimental plasmids. Again, the top panel GFP expression only continues in pike 107 while IPTG is not being exposed, and pike 105 switches off in its absence, which is the reason that they stated that pike 105 was not actually bistable. It's actually monostable. And again, here PTAC 102 is used as control. And pike 108 is used as the control for ATC, as it is induced to create GFP in ATC's presence. And the second question was, what is the effect of the inducer on the system's dynamics? The inducer essentially plays like the role of the switch between the two stable steady states. For the experimental plasmids, in this case, the inducer IPTG causes the system to be switched into what the authors refer to as the high state. In both the PTAC and pike plasmid cases, the inducer IPTG activates a promoter that in turn transcribes the repressor and GFP. The repressor expressed after IPTG induction acts on the promoter in the system, preventing expression of the second repressor. And GFP is just used as a reporter. And that's what's tracked as the y-axis on this figure, whereas time is the x-axis. When induction to the low state occurs, either through temperature or ATC as explained above, the second repressor is expressed, which in turn acts on the first promoter, shuts off its expression of GFP. This is just basically the reverse of what happens in the IPTG-induced situation. This system ensures that after induction, only one repressor will be expressed, either for as long as the system physically exists or until the plasmid is exposed to the opposite inducer. This nature is shown by the fact that controls only express GFP when they're directly being exposed to their respective inducers, while the bistable plasmids continue to express GFP after inducer exposure stops, and they only stop GFP expression when that inducer, when the opposite inducer is introduced.
So my question was for 4C. My question was, please explain how long-term stability is apparent. And as we can see in 4C, it really drives home the idea of bistability between the low GFP state and the high GFP state. In this graph, the x-axis represents time in hours, and the y-axis represents the levels of GFP expression. And the gray region represents the time when the inducer is present, whether it be IPTG, heat, or anhydride tetracycline. There are two lines on the figure that represent that are representative of PTAC, one of PTAC 117, one of the plasmids that show bistability. One represents the levels of GFP expression without the addition of the IPTG inducer, and the other line represents PTAC 117 being induced by IPTG. These results clearly show that the two-element positive feedback loop contains two stable steady states and two basins of attraction. PTAC 117 will either stay at low level GFP expression without any external cues, and PTAC 117 will stay at high levels of GFP expression with an external cue, which is representative of a biological memory unit. This again provides evidence that the system is bistable due to it having two basins of attraction and two stable phenotypes. And the top line is representative of the IPTG induced PTAC, and even after exposure to the inducer, it will still stay at high levels of GFP expression. The optional question I was asked was, what are PTAC 102 and 106, and why are these used as controls? These are used as controls because they represent a monostable system. These systems can be altered through external stimuli such as IPTG or heat. But as soon as these external factors are gone, the system will tend to go back to its singular stable steady state, which in this case is low GFP expression. These are used as controls to demonstrate the difference between monostable and bistable steady states. And that's it. Yeah.
So for subgroup 3 of paper 2, we will be explaining figures 5 and 6, and the specific questions are, do the dynamics of the toggle switch generally display robustness to noise? At which concentrations of the inducer is the toggle switch most sensitive to noise? And where does the noise sensitivity come from? So specifically for figure 5, we're looking at, or for 5A, we're looking at the dynamics of PTAC 117 toggle switch at gradually increasing concentrations of the inducer IPTG compared to the dynamics of the PTAC 102 control under the same conditions of increasing IPTG concentration. And then so in 5A, we see that PTAC 117 shows a quasi-discontinuous jump from low to high states, while PTAC 102, the control, shows a sigmoidal curve. We can see the sigmoidal curve here in like bluish purple, and the discontinuous jump of PTAC 117 in red. We also see that there's some variance around the discontinuity around after, I think it's 40 micrograms of IPTG inducer. And this is caused by the stochastic nature of gene expression. So since this specific toggle switch is dependent on the expression of a gene, of the GFP gene induced by the IPTG inducer, the variability we see with the times at which the gene would be expressed produces the variance. Next, for figure 5B, we're seeing like a clearer look of the discontinuity between the switch from low states to high states at this specific inducer concentration. And so we're just looking at the fractions of PTAC 117 cells in high state versus low state. And as you can see, there's just like less variability in it because we're looking at the fraction of cells rather than the GFP expression specifically, which removes some of the reliance on like the reliance on the measure of gene expression. And then for 5C, we're looking at scatter plots and histograms of the toggle cells at selected points about the bifurcation point. So in the last figure, they had points 2, 3A, 3B, and 4 marked that were around that 40 micromolar concentration of IPTG. And so those were selected to look further into, and we're looking at GFP fluorescence versus concentration of IPTG. And as you can see, around point 2, which was still in the low expression state, but near the bifurcation point, we're seeing a normal binomial distribution for GFP fluorescence. At the bifurcation point, which is points 3A and 3B, we're seeing a bimodal distribution, which is kind of like the fusion of two binomial distributions. And that allows us to see that there is a toggle switch mechanism happening at this point. And then this is also compared to point 4, which is in the high expression state after the toggle switch mechanism. And then we see it return to a normal binomial distribution.
All right, so in figure 6, we see in the PTAC 117 that with the switch from the low to high state, it takes a long time. We can see here the time course going from 0 to 6 hours, and then over with the transition from the high to low state, it goes very quickly, under 30 minutes. So in this part, they're asking why is this, and why the transition from the high to low state is so much faster. So in the transition from the high to low state, it relies on the gradual dilution by cell growth of the IPTG bound lac repressor, which causes the cells to switch from this low state to the GFP producing state. And this takes a lot of time. It takes about 6 hours for this to fully complete. And then on the other hand, we have with the switch from the high to low, it uses thermal destabilization of the temperature sensitive lambda repressor. So this happens very quickly, and then the repressor will no longer be working there. And then that will go from the GFP producing cells to the non-GFP producing cells. Additionally, the rate of the lac repressor synthesis is a lot higher than the lambda repressor. So that indicates that the low state is more stable. And then here for part C, you can see the switch from the low to high state with the IPTG induction, where you start out with these cells in the low state, and then over the course of 6 hours, they fully switch over to the high state.